Good morning to all. Today we will continue with the discussion on gene expression. And yesterday we discussed prokaryotic transcription in detail. Now we will continue with eukaryotic transcription and its regulation. In case of eukaryotic transcription, when we discuss about eukaryotic transcription, it is having a lot of difference with the prokaryotic system. So basically we can say that the eukaryotic transcription is actually separated by space and time. Why it is said so? In prokaryotic system, for example, we have a DNA from which the mRNA is copied during the transcription process. So this DNA is present in the cytoplasm itself. And during the synthesis of the mRNA, or during the synthesis means the mRNA synthesis is not complete yet. So even during the synthesis of mRNA, the free end of the mRNA, the free 5' or 5' end of the mRNA that is available will be bound by the protein synthesis machinery, that is the ribosomes, and it starts translation. So this translation system will not wait for the entire mRNA to be transcribed, which means transcription and translation is occurring simultaneously in a prokaryotic system. And that happens because the prokaryotic transcription and translation is occurring in the same cytoplasmic space. So it is occurring at the same time in the same place. So that's why the primary transcript or the mRNA that is produced is available for transcription, and it starts translation immediately on the available free end of the mRNA that is newly generated. So that's why there is no space and time separation in case of prokaryotic system. It occurs simultaneously on the same cytoplasmic space. Whereas in case of a eukaryotic system, we can see that there is a nuclear membrane. The nucleolus is shown here, and this is the nuclear membrane. And the DNA is present inside the nuclear membrane. And during the transcription process, this packed DNA will be unwound or unpacked, and that will be made available for transcription. And the primary transcript that is produced in a eukaryote needs to be processed in using a variety of splicing mechanisms, and the final mRNA is produced inside the nucleus. Then that that processed mRNA needs to be transported across the cytoplasm, across the nuclear membrane to the cytoplasm, where it is translated. So the translation process and transcription process is not occurring in the same space and at the same time. It is not occurring in the same space and time in a eukaryotic system, because the primary transcript is produced in the nucleolus, and the protein synthesis occurs in the cytoplasm. So it is separated by space, and it is separated by time. You can see that the primary transcript requires to be, it is not ready to be translated. It needs or it requires processing to produce the final functional mRNA. And also it requires to be transported across the nuclear membrane to the cytoplasm, where it is translated. So that's why the eukaryotic transcription process is separated by space and time, and it is not like that of the prokaryotic system, where transcription and translation occurs simultaneously in the same space of a cytoplasmic space. Okay. Now, the eukaryotic genes consist of various control elements. The control elements are the non-coding DNA that regulates the transcription by binding to certain proteins. So the control elements are DNA sequences or regulatory DNA sequences present in the gene, and they are bound by some proteins. The distal elements called enhancers are very important in eukaryotic transcription. So there are different control elements or specific sequences, control sequences in the non-coding part of the DNA, and that non-coding DNA which is associated with the genes, which is known as control elements, is required for binding of the regulatory proteins. And there are distal elements which is actually normally present upstream of the gene, and these distal elements were known as enhancers, and they are also very important in case of a eukaryotic transcription. Then there are transcription factors. The transcription factors means the general transcription factors actually they result in very low RNA production. So there are some of the proteins that is binding or interacting factors that is binding to the transcription assembly or the gene were known as general transcription factors. But the general transcription factors offers only very low rate of RNA production or transcription. But there are other specific transcription factors which are coming under the category of activators and repressors. So the activated proteins which are coming under the specific transcription factors can promote high levels of transcription. So without the activator molecules, you can expect only a very low level of transcription activity in eukaryotes. That means general transcription factors offers only low transcription rate, but with the activators or the specific transcription factors, you will have a very good quality of transcription activity or gene expression. So the activators are the proteins that stimulate transcription, and also there are some other transcription factors or specific transcription factors like repressors which will inhibit gene expression. So a gene regulation doesn't mean that it always enhances the gene expression. Sometimes it enhances the gene expression, and sometimes the gene regulation requires the stopping or suppression of the gene expression through the mediation of repressor molecules or repressor protein. Now, activators and repressors may alter chromatin structure and thereby further influencing the gene expression. So these activators and repressors are the molecules or the proteins that alter the chromatin structure to affect the transcription activity. And we will see how it occurs in the next sessions. Enhancers and silencers are position and orientation independent DNA elements or DNA sequences that stimulate or depress or repress the transcription of associated genes. So if you have a gene, for example, gene A, the gene A will have some upstream elements or some regulatory sequences associated with that one, which is a 5' untranslated regions of the gene will have some DNA sequences or DNA elements which are position and orientation independent. Position and orientation independent means sometimes it may be placed in the immediate vicinity of the promoter and operator, or sometimes it may be placed very far, means thousands of base pairs away from the functional or the gene to be transcribed or the regulatory elements. That means the position of the enhancer or silencer DNA sequence is not that much relevant. Sometimes it is very close to the gene to be transcribed, or it may be very far away from thousands of base pairs away from the gene to be transcribed. So enhancers or silencers are DNA sequences or DNA elements usually present upstream or near the 5' untranslated regions of the gene, and they usually stimulate or enhance the transcription activity with interaction with some activator proteins, or they depress or suppress the transcription activity by binding with some repressor molecules. They are also tissue specific in that they rely on tissue specific DNA binding proteins for their activities. So usually they or enhancers or silencers are tissue specific. As we discussed in one of the previous sessions, there is a tissue specific expression of the genes. For example, the transcription factors or specific transcription factors like activators or repressors were usually tissue specific. Those set of activator molecules or repressor molecules that is acting on, say, pancreatic tissue may not be available in a neuronal tissue or some other type of specific type of cells. So there are tissue specific transcription factors are available, and that tissue specific transcription factors will bind to the enhancers and silencers or the particular DNA sequence elements which is present, and the specific combination of this binding results in specific gene transcription activity. We discussed this thing in one of the previous sessions. You may refer back that one. Anyway, tissue specific activity or transcription activity is the result of specific transcription or tissue specific transcription factors or activator molecules or repressor molecules that will bind to the specific tissue specific enhancer and silencer elements which is associated with the gene. Now, sometimes a DNA element can act as either an enhancer or a silencer depending on what it is bound to. So, the enhancers, silencers are not always separate elements. Sometimes the same DNA sequence or the same DNA element can act as an enhancer or silencer, alternatively silencer, by binding with different transcription factors. So, if an activator binds to the particular sequence, it will act as an enhancer. Or if it is bound with some repressor molecule, then it will act as a silencer of the transcription activity. Okay. Here, the RNA in eukaryotic cells is synthesized by three different types of RNA polymerases. In prokaryotic system, we have only one transcription, one RNA polymerase, which is having 5 different subunits, as we discussed in yesterday's session. But in eukaryotic system, we have 3 different types of RNA polymerases: type 1, type 2, and type 3. The location of the activity of these RNA polymerase type 1 is nucleolus. The cellular transcripts that is produced by the type 1 is 18S rRNA, 5.8S rRNA, and 28S rRNA. And the RNA polymerase type 2 is active in nucleoplasm, and they produce mRNA precursors and snRNA, or mRNA precursor means the primary transcript, which needs to be processed to produce the final processed mRNA. So the mRNA precursors were coded by RNA polymerase 2, and you may note that the RNA polymerase 2 is the major RNA polymerase enzyme that is producing the mRNAs, which results in protein synthesis, and it also codes for small nuclear RNAs. Then the next category of the RNA polymerase is the RNA polymerase type 3, which is also present in the nucleoplasm, and it synthesizes tRNAs and 5S rRNAs. And all these RNAs or RNA polymerases were identified, or it is studied by analyzing the effect of alpha-amanitin on these different type of RNA polymerases. The type 1 RNA polymerase is insensitive to the alpha-amanitin, whereas it is strongly inhibited, the type 2 is strongly inhibited by that particular protein, and the type 3 RNA polymerase is inhibited only at high concentrations of alpha-amanitin. So varying responses of RNA polymerase types is present for alpha-amanitin treatment. Alpha-amanitin is actually it is produced by a poisonous mushroom, Amanita phalloides, and it binds very tightly to the RNA polymerase 2 and thereby blocks the elongation phase of the RNA synthesis. So if alpha amino is added to the system of or where transcription occurs, the RNA polymerase 2 is strongly inhibited by this particular inhibitor, that means the elongation phase. Of course, initiation occurs, but the elongation phase of the transcription is strongly inhibited or blocked by the alpha amino treatment in the case of a RNA polymerase 2, which is a major gene expression enzyme or the RNA polymerase, which is used for a synthesis of mRNA. Now the next topic is on promoters. So the promoters in eukaryotes, actually these are promoters are sequences that support initiation at normal efficiency and proper control. So these sequences or promoters support initiation at normal efficiency, and also they allow proper control of the gene expression. Promoters for RNA polymerase 2, which is the major polymerization enzyme or transcription enzyme in eukaryotic system, there we have various regions. The first region or first part of the promoter region is the initiator sequence, which is marked as INR, which is having a specific sequence, maybe a pyrimidine, two pyrimidines, then C, A, and another two pyrimidines. So that is a specific sequence for the initiator sequence, and that is considered as the start point of the transcription. So this is where the first base of the RNA is placed, starting from the initiator or initiator sequence. So that is known as a initiator region of the promoter. Now, upstream of the initiator, or towards the 5' direction of the DNA sequence, we have, or the RNA sequence, we have a Tata box. Tata box is having a specific sequence or consensus sequence of a TA, which occurs at about or in between 25 to 30 base pairs, or minus 25 to minus 30, which is present upstream of the initiator sequence or the starting point of the transcription. The second box that is present, or the third element in the promoter region, is the CAT box, and the specific sequence or consensus sequences for that CAT box is GG, CA, CC, AA, T, CT. So that is the specific or consensus sequence for the CAT box, and which is about minus approximately 80 base pairs upstream from the initiator point. We have these CAT box, and about 19 base pairs upstream we have another box which is known as a GC box, which is a GC rich box with GGG, CGG, that is a GC box sequence. So these specific sequences or specific boxes of consensus sequences at 25 to 30 and approximately 80, minus 80 and minus 90. So at these regions, the promoters, eukaryotic promoters, is having three different or distinct boxes, that is Tata box, CAT box and GC box. Then there is another octamer sequence, that is a AT-TT, GC-AT, that is another octamer sequence, which is also another upstream regulatory element or specific box that is available in the case of a prokaryotic promoter. So these are the promoter regions of a prokaryotic system. Then 1 and 2, that is the initiator and Tata box, constitute the generic promoter. So the generic promoter is the 1 and 2, that is the initiator and Tata box. Whereas CAT box and GC box were upstream, known as upstream elements, where means it is present at a distant location or somewhat distant location of minus 80 to minus 90 base pairs in the upstream direction. So 1 and 2 constitutes the generic promoter, and the generic promoter offers a very low rate of transcription. But with the CAT box and GC box, we can see that this CAT box and GC box were the upstream elements which will bound with the specific transcription factors, resulting in a structural change in the DNA sequence at that upstream part or the promoter region, resulting in a high transcription activity. So GC box and the other upstream elements were of extreme importance in transcription control because it is interacting with the specific transcription factors to enhance the transcription activity or even to suppress the transcription activity. Whereas the first two items, that is initiator sequence and data box, of course it allows transcription only at a basic rate. So they are known as a that is known as a generic promoters. Okay. So these are the different elements, that is, or elements means DNA sequences in the promoter region, data box, CAD box, and GC box. Then IG octamer, then AP1, serum response, heat shock. So there are various other specific sequences are there. The consensus sequence is also marked in the table. And on the right side, we can see that there is a lot of different specific factors or proteins which will bind to the data box. Data box is bound with a TBP or data binding protein, and CAD box is bound with another specific protein. GC box is bound with SP1, and then the other GC box, there is a different sequences of the GC box is also present, and that will be bound with SP1 or MyOD or NF1. That is a different type of factors that is binding to the specific type of GC box. Then other hormone serum response elements or heat shock protein binding sites. So that type of additional DNA sequences which may bind to specific activator molecules or suppressor molecules were present in the promoter regions. So this is a table that shows the different control elements and their consensus sequences and the corresponding protein factors, that is activators or suppressors of specific transcription factors that binds to the particular promoter. Now when we look at the overall picture of the prokaryotic promoter region, we can start with the initiator or the +1 point starting from which the transcription activity starts. So here is the transcription start point, which is a +1 point or initiator sequence. And this is the coding region or the mRNA, and this corresponds to this sequence is actually coded into the mRNA. So starting from the initial region, the mRNA is copied towards the downstream direction. And in the upstream part, you can see that the data box is present, which is normally present in between 25 to 30 base pairs or -25 to -30 base pairs upstream. Then GC or CAD box, which may be about 80 base pairs upstream, and which is also known as the proximal elements. Then there is enhancers and repressor binding elements. Then other regulatory elements which may be far apart, maybe thousands of base pair apart, upstream activation elements, etc. were present. As we discussed, the initiator element and data box or even GC, some amount of GC box, this constitutes the generic promoter region. That is the initiator and data box is the promoter region which allows a basic transcription. Whereas the promoter distant elements and also it allows the basal, this GC box or CAD box also allows basal expression of a gene. So GC box, data box and initiator sequence, these regulate the basic control, regulation of or basic expression of the concerned protein. Whereas the enhancer and repressor elements or other upstream activation sequences or distant regulatory elements were present maybe hundreds of base pairs upstream of the untranslated regions, and that type of sequences were bound with enhancers or suppressors or specific transcription factors. Enhancers and repressors are known as specific transcription factors, and it will be bound in these distal regulatory elements, and this results in regulated gene expression. So the initial part, this offers basal gene expression at low rate. So a few number of mRNAs will be produced or RNAs will be produced by the basal expression. But in regulated gene expression, if it is bound with an enhancer molecule or activator molecule to the enhancer, then it maybe 20 or 100-fold activity, transcription activity will be there. And also, if repressor molecules were bound to these distal regulatory elements, then it drastically reduces the transcription activity. So this is the overall picture of the eukaryotic promoter region. So we have to understand the promoter, basic promoter, or basal, which allows the basic transcription activity, and then this distal regulatory elements, which results in the production of a specific activity like enhancer or repressor activity. Now, the next point is on cis and trans-acting elements. Cis and trans-acting elements are the lock and key of the transcription. As in the case of prokaryotes, promoters are always on the same molecules of the DNA as the gene they regulate. And consequently, promoters are referred to as cis-acting elements. Cis-acting elements were actually associated with the gene to be translated or transcribed. So means immediately upstream of the gene to be transcribed, there will be a promoter region. So it is associated with the gene to be transcribed. So that type of elements were known as cis-acting elements. Now, the next one is, in the case of eukaryotes and their viruses also contain cis-acting elements or enhancers or repressors. And these DNA sequences, although not promoters by themselves, can enormously increase or decrease the effectiveness of the promoters. Interestingly, the position of the enhancers related to the promoter are not fixed and they can substantially vary. So, enhancers or repressors, which is known as a cis-acting elements, which will be associated with the gene to be transcribed, is present in case of some viruses also. And these type of DNA sequences, which is not as promoters, but they are like the upstream elements that regulates or which binds with the enhancer or repressor elements, which is the regulatory component present upstream. They are also a part of the cis-acting elements associated with the gene. So in the previous picture, we can see that this is the basal elements, or which allows basal expression, which is associated with the gene to be coded. So that means this is a cis-acting element. And these elements or enhancers or repressors, and this type of elements were also present in the immediate vicinity associated with the gene. Maybe it is hundreds of base pairs apart, but still it is present in the same gene, associated with the same gene to be coded. So that type of genes were known as cis-acting elements. Now, the DNA sequence of the cis-acting elements are binding sites of the proteins called transcription factors, specific transcription factors, or general transcription factors. And such a protein is sometimes called a trans-acting factor. So, the proteins that is binding to the cis-acting elements, that is the enhancers or repressors, they were known as trans-acting factors because it is coded somewhere else. So the activator molecule or the repressor molecule which will bind to the enhancer or repressor elements or the DNA sequences were produced from some other location. Okay, it is not coded by the same gene. So the activator proteins or the repressor proteins which will regulate the enhancer or repressor functions, which will be produced, that is actually produced from some other distant location, maybe from some other chromosome, and then that protein will come and bind to the enhancer regions. So that type of proteins were known as trans-acting factors because the transcription factors were not coded by the gene cluster, but it is coded from some other distant location. So that type of proteins or the transcription factors which is binding to the repressor molecules or the enhancer and repressor elements were actually known as trans-acting factors because they are coded by genes on a DNA molecule other than that containing the gene to be regulated. So it is not physically associated with the gene to be transcribed or regulated. So the enhancers or repressors, repressors are also known as silencers. These type of DNA elements or DNA sequences, they are found in a variety of locations, both upstream and downstream. So the enhancer or repressor elements or the DNA sequences that regulate the gene expression were not always present upstream, but in some cases they are present downstream of the transcription site or initiator site, or even between or in between the transcribed elements or the gene itself. So at various locations, this transcription enhancer or repressor DNA sequences can be expected. It is, or these regulatory elements were not always present upstream, but sometimes it is present downstream or even in between the gene sequence itself. Okay. And they can exert their effect when located hundreds or even thousands of base pairs away. And as in the case of the example that we seen just now, the distal regulatory elements or upstream regulatory elements were present thousands of base pairs away. So the basal elements were present very close, like Tata box or GC box, whereas the other upstream activation elements were maybe present hundreds or thousands of base pairs away to the that is in contrast to the proximal and upstream promoter elements. So these proximal and upstream promoter elements were actually the elements that contributes to the basal level of transcription, whereas the other one, that is the upstream elements, they are located hundreds of base pairs away, but still it is associated with the gene. That's why they are known as cis-acting elements, and they can exert its effects even if it is placed thousands of base pairs away from the gene to be regulated. So the next point is on upstream activation sites or UAS, which is also some distal regulatory element, which is like an enhancer or repressor elements, which is present far upstream of the gene to be regulated. There are also hormone response elements which will bind with the steroids. So in response to the steroids, usually the cell transcription or the gene expression is regulated through the mediation of some hormones. If a hormone is present, maybe gene is transcribed and the concerned protein is produced. And if the hormone stimulus or the steroid hormone is not there, then the gene is regulated and it is suppressed. So the steroid hormones will be received by the hormone receptors, which is having a DNA binding module, a hormone or steroid binding module, and a gene activation element. So that gene activation element of the hormone receptors, the hormone receptors actually, that's also discussed in one of the previous sessions. Hormone response elements will have three different regions. One is a specific DNA binding domain, and another one is a steroid binding domain, and the third one is a gene activation domain. So the hormone response elements actually they bind to the gene activation part or the enhancer regions of the gene, and these hormone response elements act as or in conjunction with the enhancers or silencers for increasing or decreasing whatever the target is. So some hormones may increase the transcription activity, or some hormones can suppress or silence the gene activity. So these are the different types of enhancer or repressor elements that is present. Now, a small number of genes lack a Tata box, and in such instances, two additional cis elements, an initiator sequence and the so-called downstream promoter element DP, direct RNA polymerase II to promoter, and in doing so provide a basal transcription starting from the correct site. So if a gene is missing the initiator sequence, then start point or the +1 point cannot be identified normally. So in such cases where the initiator sequence or the +1 sequence is missing, then the other elements like downstream promoter element or DPE is there, which supports the RNA polymerase to correctly position and start the transcription from the +1 site. Then there is another one, BRE, that is a transcription factor 2b recognition element. So B recognition element, that is BRE. Transcription factor 2b is in short marked as a B. B recognition element or transcription factor 2b recognition element is present upstream to some data containing promoters. So B recognition element is another upstream element or regulatory element where we have data containing promoter is present. Then there are different classes of promoters. In the class 1 promoters, they are not well conserved in the sequence from one species to another, but the general architecture of the promoter is well conserved. So and it consists of two elements. The class 1 promoters consists of two elements: a core element surrounding the transcription start site and an upstream element or UCE about hundreds of base pairs farther upstream. So in 100 base pair upstream, there is a UCE or upstream control element, and there is a core element that is present at the transcription start site. So that type of promoters were known as class 1 promoters, where it is having two components. One is near the transcription start site, and another one is the upstream control element, which is about hundreds of base pairs away. And the spacing between these two elements is also important in the regulation of the transcription. So the class 1 promoters is having some DNA sequences or some control elements at the transcription start site and also about 100 base pairs away. So that's a class 1 element or class 1 promoter. The class 2 promoters may contain any of these following elements. That is a data box, an upstream element, there is a GC box or CAT box, and an initiator and a downstream element. So the promoters in eukaryotes were not always in the same class. Sometimes it is using a class 1 element with some regulatory elements near the initiator sequence and 100 base pairs away regulatory elements. Or class 2 promoters which uses the data box, GC box, CAT box and initiator sequence, which is the example that is shown in the previous slide, where we have an initiator sequence, so it's a GC or CAT box and a data box. That type of promoters were known as class 2 promoters. At least one of these elements is missing in many other promoters. So you can't expect all these initiator, data box, GC box and CAT box, all the categories were present in all the genes. So sometimes maybe CAT box is present or GC box is present or data box or some of the regions or the elements may be missing in some type of a promoters in eukaryotic systems. Now you can't expect all the type of promoters in all genes. Promoters for highly expressed specialized genes tend to have data boxes. So highly expressed or more frequently expressed genes were always included with a data box. So it is that much important. But promoters for housekeeping genes tend to lack them. So housekeeping genes usually they are expressed at the basal rate, and that type of housekeeping genes which is constitutively expressed always in the same rate, which actually it is not well regulated, because in the regulated gene expression we need activators and repressors or silencers. All these things will determine whether the gene to be transcribed or not. But in the case of housekeeping gene, they are basically coding for the structural components which is required for keeping the cell alive. So such type of genes were basically transcribed at the same rate throughout the lifespan of the cell. So that type of housekeeping genes usually they tend to lack this data box. And some promoters have conserved initiators, and some promoters, including the mammalian TDT promoter, they contain only an initiator and possibly with a downstream element. So most of the genes have some kind of upstream element and may have more than one. Some may have more than one. So the concept or the basic idea is that we have various type of elements like initiator sequence, data box, GC box, CAT box, or other TDT promoter region. So various types of elements or regulatory elements are there, but all these sequences were not always present. In some cases, for example, specialized gene expression, data box is present. Housekeeping genes, data box may be absent. or some other specialized promoters. Some of the regions may be present even the initiator regions. Then class 3 promoters were for RNA polymerase 3, and that RNA polymerase 3 transcribes a set of short sequences or short genes, and the classical class 3 genes, that is the 5S RNA and tRNA and adenovirus VARNA genes, have promoters that lie completely within the genes. Okay, the internal promoter of the 5S RNA gene is split into 3 regions, that is box a short immediate intermediate element, and a box C. And another type, there is a promoters of a later 2 class, that is the tRNA and adenovirus VARNA genes. They have that part promoters were split into 2 parts, box A and box B. So different types of regions were present in the class 3 promoters. Class 3 promoters were basically used for RNA polymerase 3, and in the case of RNA polymerase 3, as we discussed, it is coding for 5S RNA, tRNA, and adenovirus VARNA, etc. And these 5S RNA coding promoter, that is having 3 distinct regions, box A short intermediate element, and box C. Box B is absent. Whereas in the other case, that's tRNA coding promoters, where we have 2 boxes, box A and box B. So different class of promoters is having different set of control elements. Now when we come to the transcription factors, they are the proteins essential for transcription initiation. Basically, there are 3 types. Basal transcription factors are present, upstream factors, and regulatory factors. In the case of these, you can see this is the proteins that is required for the initiation of transcription at the RNA polymerase 2 promoters in eukaryotes. So at the initiation stage, we have polymerase 2 that is used, then tata binding protein, then transcription factor 2A, 2B, 2E, 2F, and 2H. These are the different factors that is binding during the initiation stage. And during the elongation stage, we have various elongation factors that is used. So is a list of them, and their functions is also shown in the table. The subunit size and number of subunits present is also represented in the table. You can go through this slide from the PPT that is provided along with this video. So when we come to the different 3 classes of transcription factors in class 2 genes, here basal components or basal transcription factors, and the specific components is a tata binding protein, transcription factor 2A, 2B, 2E, 2F, and 2H. So these are all relevant for transcription initiation. In the previous slide, this is listed in the transcription initiation components. Then co-activators. Co-activators include TAFs and TBP associated factors. So TAF is actually it's an abbreviation for TBP associated factors. So TBP or tata binding protein associated factors were known as TAFs. TBP, when it is bound with the TBP associated factors, then it is known as transcription factor 2D. So transcription factor 2D is nothing but transcription tata binding protein and TBP associated factors. So this constitutes the transcription factor 2D. Then there are some activators. The activators were like SP1, ATF, CTF, AP1, etc. are activator molecules which will bind to the distal elements like the enhancer elements or repressor elements which is present upstream. So that will be bound with such specific factors or activators. The activators, they are having 3 domains. There is a DNA binding domain for specific DNA binding. Then the transcription activation domain, usually the activators were supposed to activate or increase the transcription activity. So the activators will have another domain for a transcription activation, and there is a dimerization domain for binding with the other molecules of the activators. So in the DNA binding domain of the activator, we have zinc containing modules. Usually such proteins were interacting with the DNA or binding with the DNA using zinc fingers. And the zinc fingers such as those found in transcription factor 3A and SP1, and there have two transcription factors have already been encountered. And zinc modules found in the glucocorticoid receptor and other members of this group of nuclear receptors is another case where the interaction with the zinc containing modules for DNA binding is used. So that's a zinc finger concept is there for the binding of these DNA binding proteins to the DNA. And another part is homeodomains, HDs, and these contain about 60 amino acids and resemble in structure and function to the helix to end helix of the DNA binding domains of the prokaryotic proteins such as the alpha phage repressor. So these homeodomains they are found to be they are found in a variety of activators and were originally identified in activators called homeobox proteins that regulate the development of fruit fly Drosophila. So zinc finger modules are there, homeodomain modules are there, then BZIP and BHLH motifs. There is another example where the CCATT or enhancer binding protein, the MyoD binding protein, and other eukaryotic transcription factors have a highly basic DNA binding motif linked to one or both of the protein dimerization motif known as leucine zippers and the helix loop helix HLS motif. So these are the different type of DNA binding modules or motifs which is present in such proteins. So the proteins which are zinc fingers or homeodomains or the other BZIP or BHLS motifs can bind with the DNA using these these provisions. So that's how the DNA binding domains were interacting with the DNA. So that's the DNA binding domain. So the protein or the activator molecules actually they need to bind to the specific target of the DNA using any of these combinations. That's a zinc containing or zinc fingers, homeodomains, or other motifs explained in this slide. The transcription activation domain of such activators, and in this case most activators have one or more of these domains, and then acidic domain is there. The yeast activator GAL4 typifies this group with acidic domains, and it has a 44 amino acid domain with 11 acidic amino acids. Then there is a glutamine rich domain. The activator SP1 has 2 such domains, which are about 25% glutamine. And one of these has 39 glutamines in a span of 143 amino acids. In addition, SP1 has 2 other activating domains that do not fit into any of these 3 main categories. Then there is another proline-rich domain. And the activator CTF, that's another activator, for instance, has a domain of 84 amino acids, 19 of which are proline. So there is a acidic domains present for activation, or glutamine-rich domain is present. Example is SP1, and another in case of CTF activator molecule, proline-rich domains with 19 proline molecules were available. So the first one, or in the previous slide, we discussed various provisions for binding to the or DNA binding modules which is present in the activator molecules, which will enable the concerned activator to bind to the specific target DNA. Then after binding, it needs to activate the transcription by modifying the structure of the DNA. So that activation or changing of the structure of the DNA by using such domains like acidic domains, glutamine-rich domains, or proline-rich domains in such a specific activators. So for acidic domains, the GL4 activator is the example. Glutamine-rich domain is used by the SP1 activator, and CTF activator uses a proline-rich domain. So the active, as we discussed in the previous slide, it is having 3 domains. The activator molecules is having 3 domains. DNA binding domain, single fingers or homeodomains or other specific motifs which is present. Then it is having a DNA activation domain for enhancing the transcription activity, and such activation domains is included with acidic proteins or glutamin-rich regions or proline-rich regions. Then finally, there is a dimerization domain which enables the molecules to form dimers of the activators. Okay. So this is a eukaryotic basal transcription factor complex where RNA polymerase is bound to the TATA. This is a TATA box, TATA binding protein, and TATA binding protein. This is the transcription factor 2D. 2D means a transcription factor, TATA binding protein and associated proteins, which will form a complex known as a transcription factor 2D, then 2A, 2F, 2B, 2H, 2E. All these were associated and bind to along with the or it facilitates the binding of the RNA polymerase 2. So this is about transcription factors. In most of the mammalian genes have a TATA box that is usually located about 25 to 30 base pairs upstream from the transcription start site. So TATA base is located, as we discussed, it is a 25 to 30 base pairs upstream. And the consensus sequence for the TATA box is TATA-AA, and the TATA box is bound by 34 kilodalton TATA binding protein or TBP, which in turn binds to several other proteins called TBP associated factors or TAFs. So the combination of TBP and TAF is referred to as transcription factor 2D. Binding of transcription factor 2D to the TATA box sequence is thought to represent the first step in the formation of transcription complex of the promoter. So the initiation of the transcription is actually done when the transcription factor 2D binds to the TATA box, which facilitates the binding of additional transcription factors or recruitment of the additional transcription factors and the RNA polymerase. So this is another illustration where the TATA box is bound with transcription factor 2D. As I mentioned, it is a combination of a transcription factor, TBP, that's the TATA binding protein, and TBP associated factors or proteins. So that complex is known as the transcription factor 2D. It is binding to the TATA box. And SP1 is the activator molecule that bind to the GC box. And CTF is another activator molecule which we discussed as maybe proline-rich. That CTF factor is binding to the CAT box. And then another SP1 is binding to the GC box here. And you may also note that the GC box may have different orientation. This GC box is having this orientation, and whereas this one is having the other orientation. So these are all, this constitutes the proximal upstream activation elements. Assembly of the RNA polymerase and transcription factors at a promoter is described in this slide. So the formation of a closed complex like that of the prokaryotic transcription, it is done by the TATA binding protein that binds to the TATA box. So TBP is bound in turn by the transcription factor 2B. Then transcription factor 2A can, although it is not essential, it can stabilize the TBP binding. So the TBP is not always bound as such. TBP will bind with the TBP bind or associated factors or TAFs. That is a transcription factor 2D. TBP plus TAF is transcription factor 2D. So once the transcription factor 2D binds, then that bound 2D is stabilized by transcription factor 2B. Transcription factor 2B. Sorry, it is 2A. 2A binding stabilizes the transcription factor 2B and TBP complex. The transcription factor 2B TBP complex is next bound to another complex consisting of a 2F, polymerase 2 and other associated factors. So the basic concept is that the initially TBP or TATA binding protein binds to the TATA box, And that TBP is also associated with the TBP associated proteins. So entire molecule is known as transcription factor 2D. Then it recruits 2A, then 2F, and other after 2F there is RNA polymerase 2. All these additional proteins will be recruited, and finally we have 2E and 2H, transcription factor 2E and 2H bind to create a closed complex. So the closed complex in eukaryotic initiation occurs by binding of these factors in the sequence. TBP with the TAF or TBP associated factors, which is known as transcription 2D binds first, then transcription factor 2A stabilizes that, then transcription factor 2F binds, then transcription factor 2F recruits or this complex recruits RNA polymerase 2, then finally transcription factor 2E, 2H, etc. binds, and that creates or this entire complex creates the closed complex. Then transcription factor 2H has a DNA helicase activity that promotes the unbinding of the DNA near the transcription start site. So once the closed complex is formed, the 2H is, with its helicase activity unbinds the DNA, thereby creating the open complex. So the closed promoter complex initially forms, then 2H with its helicase activity unbinds the DNA, forming the open promoter complex. So this is the diagram. This is the Tata box, initiator sequence, TBP along with the transcription associated factors and transcription factor 2A that binds, and 2F binds with the RNA polymerase, 2E and 2H binds. 2H is having the helicase activity. So the entire molecules, once it is bound, it is known as closed complex. So the 2H with its helicase activity unwinds the DNA strand, and then it forms the open complex. Then the phosphorylation of the RNA polymerase 2 and initiation and placement of a few base pairs occurs. Then elongation starts, and leaving all these things behind, the RNA polymerase moves starting from the initiator sequence. Then finally 2H and 2E and 2H is relieved, and elongation continues, and finally it reaches the termination, and then all RNA polymerase and other factors were recycled and reused in the next cycle of transcription. So this is a transcription of the gene that is in the initiation of eukaryotic system. Here the regulation of initiation is done by various activator proteins. The activator proteins were binding to the enhancer sequences or the distal control elements, as we mentioned in the previous session. So the activator proteins bind to the distal control elements or the enhancer elements or DNA elements, and then it activates the DNA transcription. The DNA binding, the DNA bending proteins, these are the DNA bending proteins which will bound to the activators. So these bending proteins create a structural change or a bending of the DNA, which brings the enhancer sequence close to the promoter region. So these bending proteins which bends the DNA, and the enhancer elements with its activators were brought to the vicinity of the promoter Tata box. So the other mediators, transcription activators or the co-activators, etc., will interact between these elements, forming a complex, and the RNA polymerase is also recruited. So this complex forms the closed initiation complex or closed promoter complex. So the activators binds to certain general transcription factors and mediator proteins, helping them to form an active transcription initiation complex on the promoter. So now, once it is formed, the helicase activity of the transcription factor 2H unwinds the DNA to form the open promoter complex, and then leaving behind all these additional proteins, the RNA polymerase move forward to the elongation stage. So this is another representation where different proteins in different combinations were binding. So it is not always in a particular sequence. Sometimes say the CTF, this is a tata binding protein, and our basal complex, etc. is shown here. TAF, TBP, transcription TBP associated factors, TAFs are binding here. And then finally it bends by binding to CTF and cat box. So this cat box is this DNA bending is finally done by incorporation of the CTF and attachment of the cat box. Or in some other cases, we can see that initially the CTF is bound, and from the CTF the TBP is recruited and the basal complex is formed. Then finally it is binding to the tata box. So in any direction the complex binds, and with the interaction of all these proteins, the cat box is brought in the vicinity of the tata box. So the DNA forms a loop, and that change in the structure results in the activation of the transcription. So this bending or this structural change in the DNA promotes the transcription activity. So these are some of the okay. Now the elongation, termination and release stages. Transcription factor 2F remains associated with the RNA polymerase throughout the elongation. The activity of the RNA polymerase is greatly enhanced by the proteins called elongation factors. Elongation factors, some bound to the phosphorylated CTDs, carboxy terminal domains, suppress pausing during the transcription, and also coordinate interaction between the protein complexes involved in the post-transcriptional processing of the mRNAs. Once the RNA transcript is completed, the transcription is terminated. The RNA polymerase 2 is dephosphorylated and recycled, ready to initiate another transcription cycle. So the RNA polymerase, once it is dephosphorylated, even RNA polymerase and other factors can be recycled, and they will be used for another cycle of transcription. Then some specific signals regulate the transcription termination. The signals for the termination of the transcription by the eukaryotic DNA RNA polymerase 2 are very poorly understood. However, it appears that the termination signal exists far downstream of the coding sequence of the eukaryotic genes. So the termination sequence is generally, it is expected to be present downstream, even after the coding region of the genes. Example, transcription termination signal of mouse beta globin occurs at several positions about 1,000 to 20,000 bases beyond the site of the poly A tail. So even downstream, even after the poly A site and the transcription termination site, and even 1,000 base pairs downstream of that poly A tail, there may be multiple termination signals were present in beta globin gene. After the RNA polymerase 2 has traveled the region of the transcription unit encoding the 3' end of the transcript. So starting from the initiator region, it forms the open complex, then closed complex, then open complex, then elongation starts, and after the elongation, it reaches a termination region where we are expecting a poly A signal, right? Polyadenylation signal is there, and that is present near the 3' end. So that signal is AAU-AAA. So that is the consensus sequence of which is present at the 3' end of the mRNA. So by recognizing this signal, we discussed when we learned that polyadenylation in the RNA splicing process and RNA processing, we already come across the polyadenylation process in which some specific sequence or polyadenylation signal is present at the 3' end. That is the AAU-AAA. So once it passes, this RNA polymerase 2 passes that polyadenylation signal which is present at the 3' end. Then and there is an RNA endonuclease activity, and that RNA endonuclease cleaves the primary transcript at that position, and the RNA primary transcript will be subjected for RNA processing and poly A tail addition. So the transcription termination occurs when that RNA polymerase transcribes this polyadenylation signal, and then the RNA endonuclease cleaves the RNA and it releases the primary transcript, and that primary transcript will be subjected for a further splicing in the eukaryotic system. Finally, this newly formed 3' terminal is polyadenylated by in the nucleoplasm in the process of splicing, as described below. So now we are almost coming to the end of this transcription, eukaryotic transcription. But let's discuss, or let's compare some of the facts that is seen in prokaryotic and eukaryotic transcriptions. First of all, transcription and translation are continuous process and occur simultaneously in the cytoplasm, because it is occurring in the same space and time in prokaryotes. Whereas these are two separate processes occurring at two different locations in eukaryotic system. So they are two separate processes, and transcription occurs in the nucleus, where translation occurs in the cytoplasm at a different time in eukaryotes. Transcription initiation machinery is simple, since DNA is not associated with any histone proteins. So the transcription machinery is simple. Just once the transcription factors or the RNA polymerase binds to the DNA, it can immediately transcribe. So always it is in a positively activated state. So or genes were always active for transcription in case of a prokaryotic system. There is no histone proteins. In case of a eukaryote, the transcription initiation machinery is very complex, since the genetic material is associated with histone proteins and tightly packed. So initiation requires activation. Normal state of the eukaryotic gene is suppressed or inactive, which requires a complex protein machinery and activation system for transcription activity or gene expression. Only one type of RNA polymerase enzyme, which synthesizes all type of RNA in the cell, and mRNA, rRNA, tRNA, all these were coded by or transcribed by the same RNA polymerase enzyme in prokaryotes. Whereas we have three different RNA polymerases in the eukaryotic system, and the RNA polymerase 1 for rRNA synthesis, RNA polymerase 2 for mRNA synthesis, and RNA polymerase 3 for tRNA and 5S RNA synthesis. We already discussed that one. Now, RNA polymerase with 5 subunits: alpha 2, beta, beta prime, and omega. And also there is another sigma factor that is involved, we know. So such a 5 subunit RNA polymerase is present in prokaryotes, and the functional RNA polymerase is alpha 2, beta, beta prime, and omega along with the sigma. Now, RNA polymerase 1 with the 14 subunits is present in for RNA in case of a eukaryotic system, and RNA polymerase 2 with the 10 to 12 subunits. Then RNA polymerase 3 with there is some error. There is a RNA polymerase 3 is with the 12 subunits. Now, sigma factor is present in prokaryotes, which is essential for transcription initiation and specific binding of the core enzyme or the holoenzyme to the RNA transcription initiation site. The sigma is absent and it is not required for transcription initiation, and initiation of transcription is facilitated by the initiation factors in case of eukaryotes. RNA polymerase can recognize and bind to the promoter region with the help of a sigma factor. The RNA polymerase cannot recognize the promoter region directly unless the promoter is preoccupied by the necessary transcription initiation factors in eukaryotes. Promoter region always located upstream to the start site in prokaryotes. Promoter region usually located upstream to the start site, but rarely there are cases in which the RNA polymerase 3, for example, Promoter is located downstream of the start site. So that is not present in prokaryotes. Always the promoter region is upstream of the start site in case of prokaryotes. But at least in some rare cases, eukaryotic system may have downstream promoters. The promoter region contains Pribnow box at -10 position. The Tata box and Cat box are absent in the promoters of prokaryotes. Whereas the promoter region contains a Tata box, which is at about 25 to 30 base pair length, minus 25 to 30, and there is a Cat box and a GC box. All these are present in eukaryotes, and Pribnow box at -10 is absent in case of eukaryotes. The termination of transcription is done either by row-dependent mechanism or row-independent mechanism in prokaryotes. A termination mechanism of transcription is not completely known, but it may be directly by the poly-A signal or the concerned RNA endonuclease which is used, which will cleave near the polyadenylation signal and release the RNA. So that mechanism is not clear. Usually, there is a post-transcriptional modification of the primary there is no post-transcriptional modification that is required in the for the primary transcript in prokaryotes, because the RNA is ready to use, ready to be translated, and even before complete synthesis of the RNA, the translation starts. Whereas in the primary transcript in the eukaryotes requires a post-transcriptional modifications, RNA splicing or RNA editing and removal of the introns, joining of the exons, and transportation across the cytoplasm. Everything is required in case of a eukaryotic system. Then RNA capping is absent and mRNA is devoid of 5' guanosine caps. RNA capping is present in case of eukaryotes and 5' cap is added in case of a eukaryotic mRNAs, not in prokaryotes. Poly-A tailing of the mRNA is present poly-A tailing is absent in prokaryotes and poly-A tailing is present in case of eukaryotic mRNAs. Then introns are absent in prokaryotes, whereas introns are present in the primary transcript, which needs to be removed by splicing in eukaryotes. Splicing of mRNA is absent since introns are absent. Splicing is present in case of eukaryotes, and that introns in primary transcript are removed and the exons are joined by the variety of splicing mechanisms. Okay. Now, genes usually are polycystonic in prokaryotic system, and hence a single transcript may contain sequences for many peptides, just like that of the lac operon. And the genes are monocystonic usually in case of eukaryotic system, and the single transcript code for only one polypeptide in eukaryotes. Then SD sequences or Shine-Delgarno sequences are present about 8 nucleotides upstream of the start codon in mRNA, and the Shine-Delgarno sequences act as the ribosome binding site in prokaryotic mRNAs, whereas the Shine-Delgarno sequences is absent in mRNA it's absent in case of eukaryotes. So this marked as prokaryotes. You may correct it as the Shine-Delgarno sequence is absent in mRNA of eukaryotes. And the initiation, correct positioning of the initiation point of the eukaryotic translation is done by 5' caps. Whereas the Shine-Delgarno sequence in prokaryotic mRNA plays a significant role in correct positioning of the initiation point. Okay, thank you. This is all about eukaryotic transcription. Okay, thank you.